Hi everyone. So welcome to the MZmine workshop. I hope you're in the right place. My name is Tomáš Pluskal and I'm a group leader at IOCB Prague. Before I start with the presentation, two important announcements. All of you should have received a link to Google Doc with the instructions where to download the software and the dataset for this workshop. If you haven't received the link, it's under this QR code, so you can take a picture. Okay. Second announcement, we have a very cool MZmine stickers, which are placed here at the front desk and also at the entrance. So whoever wants to have a nice MZmine sticker for their laptop or whatever, you can grab one. And now I'm going to start talking about what is MZmine. Does everybody here not know what is MZmine? Okay, that slide is for those people, so it's fine. Well, MZmine is a software to process mass spec data. So you start with your raw data, and you want to turn that raw data into something meaningful, something that's interpretable. Okay, so this was all the introduction. Now, as you probably know, MZmine has quite a bit of history around it. We actually just celebrated the 20 years anniversary of this project, and that you can see from these publications in 2006 about MZmine first generation, 2010 about the second generation, which has been cited quite many times, and last year we published a paper about MZmine 3 in Nature Biotech. You can also see the progress of MZmine in this chart, which shows the number of lines of code over time. And you can see it's steadily increasing and actually picking up pace in the last 5 years, where there has been a lot of development. And this has culminated with the paper on MZmine 3 last year. There are many people who contributed to the code base. We have a lot of contributions from many different labs, and there are many users, of course, many downloads of the software. So what I'm trying to show with this slide is that MZmine has been quite popular and quite a big success in academia. Also, this year we published a protocol paper in Nature Protocols, where you can see the details and instructions for all the workflows for processing LC-MS data, GC-MS data, and imaging data in MZmine. So if you haven't seen this yet, you can take a look. That said, I mentioned that MZmine has been quite a big success in academia, and so we as the main developers of MZmine have decided to also try to push MZmine into the professional sort of commercial domain, pharma, biotech companies, and so on. So earlier this year, we started a company that's called MZIO, and so from now on, MZmine is going to be not only a community project that's developed by academic labs, which it remains, but it's also going to be a professional project that's developed by a commercial company and being offered with all the requirements of these professional customers, such as support and documentation and so on, to the pharma and biotech industry. So this is our team, and you will see some of the people speaking here. And with this change, we also released MZmine 4. So there's been quite quick development. We don't call it MZmine 4 anymore. We just call it MZmine from now on because we assume the version numbers are going to change very often. And so I just want to show you a few slides on what is new in this recent development in MZmine. So we have the MZwizard, where you can quickly set up workflows depending on the type of the instrument and the type of analysis that you're doing. And you can quickly create batch files that process all your data. We have interactive molecular networking, where you can give context to your data sets. You can look for your molecules. You can overlay the annotations over your molecular networks, and you can find related molecules. You can process imaging data, where you can see the extracted images and, again, link them to your LC-MS data and use them with the context of molecular networking and such. We have an updated module for lipid structure prediction from the MS data. So you can use MZmine for lipidomics. We have a better support for GC-MS data. We have new algorithms for spectral deconvolution in GC-MS. And we have a new module for statistics, so you can see all your statistical outputs in one place directly in MZmine and linked to your raw data, so you don't have to you don't have to upload your data to Metabolomics and other tools anymore. And with this is the end of my presentation. I just wanted to highlight that you can see how popular MZmine is from the number of abstracts at this conference that actually mention MZmine, not to mention this workshop, of course. And thank you very much. With this, I would like to give word to Robin, who is going to lead on with the first talk about MZmine data processing and also with the hands-on session. So go ahead, Robin.
Thank you. So thanks very much, Tomáš. And yeah, you already saw there was some presentations and some posters that we mentioned, and that was only the ones that actually mention MZmine in the abstract. So if you're also using MZmine, feel free to, like, on social media, just ping us with @MZmine_project, and then your poster number or something, and we can then kind of like push it also on social media. And before we jump into the hands-on session, so very important, I hope that all of you have installed MZmine. If you don't, you can also follow along and just watch and kind of take it as a live session. But if you have any issues installing MZmine or anything, there's Corinna and Tito and other people. You can just point your fingers to them, and they might just come to you and help you. Okay? And yeah, also maybe already log in to the Metabolomics 2024 Wi-Fi. It seems to be open, and if there's a password, it's Osaka 2024 also. So for the first introduction, before we jump into the data processing, we already saw that MZmine incorporates all kinds of MS data, and this is also our goal. Like, we try to build one platform that can handle any kind of mass spec data, and the good thing is we always have the same kind of detector, right? Like, we're detecting mass to charge ratios, or at least we get spectra in the end after some processing. And then we can extract extracted ion chromatograms. We can do this also with eye mobility data, also with imaging. And so the whole workflow might look something like this, where we do some spectral processing in the beginning. We can import some native data formats from the vendors. And we're currently working with all the different vendors to also get more support for data formats. But typically we say, okay, convert your data to MZML, which is the latest standard format, open standard format. Or if you're doing a TIMS data analysis, just use the native TIMS data, because it's getting too big if you convert it. Then we do spectral processing. We try to find the same MZs across the whole retention time range and create extracted ion chromatograms. And then we try to separate those into features. And a feature, because we're doing mass spec, is basically an ion, hopefully, or maybe noise, right? But it's an MZ that's defining the feature, and then we have some additional metadata like the retention time, peak shape, maybe CCS, eye mobility, and all other properties that we measure. And because it's a modular system, we can then easily plug in some more modules for eye mobility spectrometry. And we can also plug in some modules for ion identity networking, where we try to find all the different ion adducts of the same molecule. And we can also do downstream analysis in molecular networking we're going to look at, and statistical analysis and so on. But the main goal of the first approach is that we generate an aligned feature list. And the alignment really happens on all the different properties that we have in your data set. So maybe eye mobility, MZ, and retention time. And because in the end, the whole modular workflow that we set up is usually quite complex. Like in the early days, maybe some of you are still using MZmine 2. Maybe hands up who's still using MZmine 2. Okay, 1, 2. Yeah, don't be shy. There's many people I know. I really say, like, you should really try MZmine 4 now, because it's I think it's so much nicer how everything is set up. Because setting up this workflow can be quite stressful, and now with the MZwizard, you can really, like in your lab, just connect the different technologies together. So you have LC, maybe flow injection, maybe MALDI, then TIMS, no IMS, and then a different mass spectrometer, and also the workflow. Later we're going to see from Corinna also library generation or data-dependent acquisition. So there's different ways we can analyze data. And before we jump into the interface, this one I'm going to show you directly. I also going to introduce you to molecular networking, because this is like our goal for today. We have a data-dependent acquisition, non-target LC-MS data set that we want to analyze and then cluster the chemical space. So we want to try to see if there's chemicals that are similar in our own data set and then cluster them in a network. And this has been popularized for small molecules by the GMPS online platform, and there's a strong collaboration between the two groups, as I've been also visiting Peter's lab, and we've been working on this for a long time. Louis, sadly, he cannot be here today, but he's going to be here from tomorrow. And then together with Daniel and Ming, we really invented this like feature-based molecular networking, where we represent features as nodes in a network. And every time when you then see those nodes in the network, just remember that they have a peak shape, they have a retention time, MZ, and MSMS spectrum. So this is very important, that we need the MSMS fragmentation spectrum to then calculate the spectral similarity between those spectra in our data set. It's very similar to spectral library matching, and just that we also account for modifications. And then in the second step, we also do spectral library matching to annotate compounds. You could also use some in silico tools and then map all the annotations back into the network. And just with the network again, so spectral library matching, we really just go for the exact matches, right? And if we now have an analog of one compound, so we have a methyl group, if we fragment both compounds, like either we lose the modification and we generate the same fragments, or, of course, we have the same neutral losses, right? And this is what the modification-aware cosine similarity here connects. And if we do this, we're going to end up with those networks. Some of them look very cluttered, like here. But if we zoom in, really, some of those nodes are annotated by spectral library matching, and we can really propagate those annotations from one node to another node. And very often, it's just a single modification, like here for the alkaloids. Or if we have some like small peptides, we can also already imagine like many different amino acid substitutions that also are going to have very similar fragmentation. And also for polyphenolic glucosides, you can already imagine there's two compound classes being mixed together, and both of them can be very diverse. And then molecular networking usually captures this quite nicely. And in the second layer, we're also going to overlay information that we get from MS1. So we're going to look at the chromatographic peak shapes. We also look at the MZ differences between co-eluting features. And then we can connect all the in-source fragments, like water losses, and also adducts and multimers together. And if we do this, we're going to see in MZmine ion density molecular networking, here a data set together with Annika, where she only got 2 annotations. And then in a circle, you really have like one molecule. So we have multiple ion adducts of the same molecule in a circle, connected all with the same retention time, same peak shape. We all know this now. And then with the blue edges, the MS2 similarity, we then connect analog compounds. And just by propagating like a mass difference of 16, of course, might be just another hydroxy group here, you can propagate this information, then isolate, and then, of course, identify those, for example, with NMR. And the great thing here is really that with all the different ions that we can detect, we can then also get some additional information. And here, for example, there's one information that on the left side, we have one hydroxy group more, and all those arrows are 1 water loss, 2 and 3 water losses, and on the other side, we only have 2 water losses. And so if we have 3 water losses, there is also one proof that it might just be another hydroxy group on an aliphatic ring. And now we can jump into MZmine. So I hope everyone who wants to follow along has MZmine downloaded. And you can just open MZmine. And okay, I will try to make it bigger, actually. Did not think about this. Okay, that's huge. Okay, so try to open MZmine, and if you have any issues, there's some people around you can look for and then point to them. Otherwise, just follow along. And first thing that we have to do is that we click on the user icon here, or on the top there is users, and then manage users. And in here, you can click on sign in or sign up. This might be already coming on your side. Oh, nice. Okay. Seems that there's a proxy needed. Okay, I should have just sent you the user. Okay, seems like the Wi-Fi needs a proxy, which I don't have. So, sorry? At least it says, at least on my side it doesn't work. Does it work for anyone else clicking on login? Yes? Oh, actually maybe it works clicking also on this one. See? No. Yeah. Metabolomics 2024. Sorry? Ah, okay. So seems like Wi-Fi is down. I can open Drive. Yeah, it doesn't work. Okay, so the Wi-Fi is just down. So it's not a proxy that is needed. I mean, we're going to see if the Wi-Fi comes back up. Otherwise, it's going to be, for everyone who has already logged in, it's going to be hands-on. Everyone else would then look at each other. And we're also going to just have a very like short live hands-on. So, login also? But the login doesn't work, right? Because at least for me it doesn't.
I mean, can we try internet? If
Internet was just working for a second there.
I cannot connect to MZio.io. It's weird. Oh, now it works.
Okay, so think there's a... Okay, let me see. ATC client. This one? Okay. And this is for everyone or just for me?
これはね、皆さんこの会場の方全員使えるんですか？Okay, free Wi-Fi for このホール全体、この会場全体の Okay.
So everyone.
For conference area.
Okay.
Everyone.
Okay.
Can use it.
Okay. Let me see once more. No. So it was 0.666.5006, and it's ATC client.
Not underscore, but
Yeah. Not underscore.
just say.
Very nice.
Or,
Okay.
Yeah.
Okay. Thank you so much.
One question for
Yeah.
all people with the sign in. There stands like something with proxy. Is it
Yeah,
also
yeah, yeah.
related with the internet?
Yeah. So
So you make an announcement.
yeah, we'll make an announcement.
I asked her to stay here for a while.
I can.
We can just check if everyone get it.
So, okay. Okay, so we figured it out. There's another Wi-Fi that actually works better. And you can search for ATC minus client, so ATC dash client in Wi-Fi. I also put the information in the Google Doc if you have the link. So ATC client, and the password is here, this number. Okay, so at least this one seems to work more stably, so it should be, at least works on my side as well. Yes, I will make it bigger as well. Okay, so different Wi-Fi should maybe solve the issue. So please try to log in here. Also, if the issue remains in this drive, there's another link now, and on this link you will also find a file, a user file that you can just drag and drop into MZmine if the login doesn't work. You can just download this file and basically drag and drop it into MZmine, and then you're good to go, also offline later on. Okay, so
Wi-Fi is working with everyone here.
yeah, so anyone still has issues, then just point up, and there's some people that are able to help.
Can you give us like 3 minutes to settle?
Yeah? Yeah. No worries. And also for anyone else who still does not have the Google Doc, there's the code that you can scan. So you can take a picture and just scan it. It's the same link that you should have gotten by email if you were already registered for the workshop. Yes, so, yeah, thank you. Thank you so much. Yeah, we will solve it together. Okay, so I hope that everyone is now able to either open MZmine, or if you really struggle, there's also maybe a person next to you that's also going through the whole workflow, and I will also go through the whole workflow, so you can also just have it as a live demo. But in MZmine, in MZmine now, when you open MZmine, you will see this Welcome to MZmine screen. And you can click on the user and then on Sign in or Sign up. Oh, yeah. Okay. Okay, it seems like sometimes it seems like the Wi-Fi might struggle because of the number of people. So, okay. So if you have a user, of course you can, if you reach this like sign in, sign up, so users, and then sign in, sign up, or manage users. If you reach this screen, you can type in your username and password and log in. Otherwise, you can click on register down at the bottom. And we only ask for very limited information, username, password, and so on. Please use your institutional email address when you sign up, because then you will automatically get like an academic license. Otherwise, you also directly get like a trial license for MZmine. And then you accept the conditions and click on register. You will then receive an email where you have to verify that this email actually belongs to you, and you should then automatically get your user downloaded. If anyone has issues with this, so you can try this now, but if anyone has issues with this, you can just download the user file, and it's named workshop user. I cannot zoom in here, but so workshop.MZuser. And I can just drag and drop this into MZmine. And if I do this, it will then basically add the user to MZmine. Okay, so this is another chance, like another thing you can do. Just from here, download the user file, and then just drag and drop the file into MZmine, and you should be all good to go. And in the end, you should see that in the users, now you have a user that's actually selected and that you can start just processing. It's a good thing for us also because before we didn't know like how many users we actually have, and for applying for funding and those kind of things, it's very good that now we actually know that MZmine is used worldwide. I mean, we had some estimate that was used worldwide, but now we actually see that like in like all across the world, actually people are using it. Okay, so everyone was able to start. Otherwise, there's Corinna and Roman and Tito. And what we can start with is if you download the data, we can go to the data folder, and you can just drag and drop all the MZML files into MZmine. Yeah, so we also send you a link for some data. If you don't have it, I think it's the best if you just look and just follow me here. But we can just drag and drop all the files, all the MZML files in here. And then we're already done like importing all the MZML files. Those are some microbial extracts, so LC MSMS data acquired with data-dependent acquisition on an Orbitrap instrument. But this is also very much applicable to any other setup. And now you can just double click on one of those files, and you should see that now everything like opens up, and you should, I'm actually going to switch to light mode because I think it's maybe easier to see, right? Contrast is higher. Great. So if you drag and drop the files in, you should see that now you have the files. I can just double click on one of the files, and then I can see the chromatogram. And if I click into the chromatogram, maybe okay, so, and if you click into the chromatogram, you can then also extract the scan at this exact moment. and you're going to see it on the other side. So I can just zoom in here, and then we're going to see the mass spectrum. And this is actually kind of a good sanity check, because I can see now it's a centroid data set, so it's already centroided. And from here, I know that our processing does not need to do centroiding in the first step. If you do have profile data, you have to make sure that you're centroiding the data then in MZmine or during the conversion. Another thing that you can do is, like, if you see this check mark here on the top with the raw data overview, you can always select multiple samples, and then you can basically compare them in one view. So you're going to see all the extracted actually base peak chromatograms here. You can see all the base peak chromatograms. I can click anywhere and then see the spectrum of all the 3 samples just overlaid. And maybe that there's a little bit of a difference in the MZ. So this is, of course, just a way to review your data, but then setting up the processing, we're going to go for the Processing Wizard, and it's here on the top, Processing Wizard. And here is really like where we put together our workflows. So please open the Processing Wizard, and I already have some presets. And the whole idea is that when you buy, for example, a new machine, or you develop a new method for your machine, you might be just saving, for example, all the MS parameters here and save them, and then you can just plug in together your reverse phase, HPLC plus Orbitrap dataset. You can just plug in together the settings for both instruments. And I can load a preset here. And the only thing that you have to do now here is we also have to mention all the file names here that we want to process. In the beginning, it's sometimes good to just use like a smaller set. If you have like hundreds of samples, maybe you only use like 10 of each different batch and then put them together and process a smaller data set first. Optimize your parameters, and then in the end, basically process everything. But for this, you can also just drag and drop all the MZML files from the Explorer. We can just drag and drop it in here. Or what is also quite easy, you can just say all .mzml files. And we select the data folder. And then it's also going to pick all the MZml files. It's sometimes quite nice if you can even say all in subfolder, and then you can even search in your data folder for all the MZml files that you have, and then it's going to basically show you like how much data you're actually having on your drive. Then there's the metadata file, and this is only important if you want to do statistical analysis here. So you can just click on select, and also in the data folder we shared the metadata_pa.tsv. And this file just contains some information about like the samples and then which conditions they were acquired for. Yeah, so it was microbial strains, so it's different mutant strain and wild type, and then controls and blanks. Then we did UHPLC, and I'm not going to describe all the like optimization of the parameters too much here. We can maybe have that a little bit in the questions later, what you're interested in. We also had done some workshops before, and there's some videos online where we had like a whole day basically describing all the processing optimization. But for UHPLC, yeah, there's some questions here in front. So for UHPLC here, we can set up, for example, to crop the retention time, and I highly recommend that, because in the beginning, sometimes, of course, you have a dip because you're switching from spraying into the diluting into the waste. You're starting to spray the sample. So maybe we want to cut off a little bit in the beginning, and we can also cut the chromatogram in the end. We can look at the raw data overview here. And in the raw data overview, you're going to see that it's pretty much this. Like in the beginning, we're having no signal. And then once we actually divert into spraying into the instrument, we get this little peak here, which we cut out. We could now also cut out, in the end, if we have some like cleaning step, we could also cut this out, but it's not very needed for small data sets. Then the rest we can just leave with the preset. So just make sure that you set it to UHPLC here on the top. So you're just changing to UHPLC on top, Orbitrap, and DDA. And then if you change it to Orbitrap, you're going to see the Orbitrap settings, which are a little bit different to a QTOF. And now you might say, but wait, there's so many different QTOFs. How can you have like one setting that fits them all? We don't, right? And the really important thing is that you have to optimize your data processing workflow for your instrument, but also your method. Because the noise levels very much depend on your measuring parameters, right? And so if you change to Orbitrap, there's the factor of the lowest mass detection step that we're going to do. And this one is, I would say, quite universal. It really works well for any data set with Orbitrap. And you can just increase the factor. It's kind of like a noise level. You can increase it if you have a very large data set. And if you have a small data set, you can basically set it to nothing, and it's still going to work. And we can increase the minimum feature height to 5 E5. That's just for this data set now. And that's it, basically. So there are some filters which you don't have to set now. And the only thing that we still going to set here is now the spectral library files. So in case you have some spectral library files on your laptop, there was also one that we supplied from Mass Bank of North America because it's quite a medium-sized spectral library. You can just click on Select Files. And in the data folder, you should see that there's one .json file. .json is a very general format, and this is the JSON format from Mass Bank of North America that we support here, but also from GMPS, from MZmine, so there's multiple different library formats. MSP and MGF as well, so MSP, for example, from NIST search. So we select this file here, and you should see the full path in annotations. And then, did everyone get to this point? Everyone who wanted to follow? Okay. And then the last step before we finally get some processing done and can look at it is the data-dependent acquisition, is the workflow. So there's a bunch of different workflows here, and if you have some ideas for new workflows, we are very eager to finding some ideas to put more workflows and more presets into this wizard to make it easy for everyone to reproduce. But here we're going to use the data-dependent acquisition workflow, and the only thing you have to set up is in the export path, please select a full path. So you click on Select here on the side. We can create a new folder and, for example, just call it Results. And it's then important, or at least good, on the bottom, we could then say Metabolomics Workshop Results or something like this. So we just give it a base file name. So I just typed in any base file name in the bottom, and we're going to produce many different resulting files. So we're going to export the feature list. We're going to export MS2 spectra for GMPS, for Sirius, and for other data processing tools. And for this, we really just need one file name, and then we're going to add a lot of different suffixes and extensions to it. So everyone managed to get here? Please, someone doesn't manage, wanted to follow, but it was too fast. Just put your hands up. So one person, yeah. Yeah. So they might be a little bit different from the preset, but it's totally fine to use the presets for now. So typically you would want to, of course, optimize depending on your peak width. You would optimize the parameters, but that we can see afterwards how you would actually do this. So I don't want you to put in too many numbers here. And so the presets work for this data set. And then it's very important, UHPLC, we don't have any IMS, so we don't set it up here. We have an Orbitrap instrument and data-dependent acquisition. And spectral library is quite nice. If you have it, you can just drop them in here in this annotation spectral libraries field. And the export path. Just any folder and a base file name without any extension. And then, if you have this preset, we could now just say save, and we can save this wizard preset. This is quite nice, and you can even like save, for example, just the sample introduction. So we could save this UHPLC reversed phase 10-minute gradient. We could save it, and then later we can just easily retrieve just the parameters for the UHPLC and plug them together with a different mass spectrometer. And when we want to process now, we click on Create Batch. So you click on Create Batch.
Sorry,
Yeah?
local presets is grayed out in my case. Is that normal?
What is... sorry.
Local presets, you have a, yeah, that's grayed out in my case.
Yeah, local presets is grayed out if you don't have any local presets. So in the future, we want to build up like a database of presets from different people. Like if you publish your paper, you should be able to also basically upload your preset somewhere, and then we're going to find it and maybe add it. But if you save a preset, so this one we could now call it like flow, like it's a whole workflow. So like workflow, metabolomics, like this, and OK. And then next time when we open the processing wizard, it's actually going to show up here on the bottom. Workflow, metabolomics. And also if you restart MZmine, it's going to be very easy for your group even, like for the whole group, to create presets and then say, okay, this is my HPLC method, this is my GC method, and then my QTOF, my Orbitrap, and we just mix them together. Yeah. And so from here, create batch, and you're going to see this from MZmine 2 even. It looks quite similar still. We have this batch queue, and from here, I'm not going to describe it much. We're just going to click OK and run all the steps. So, and I actually forgot something, so I'm going to... You don't have to stop it. I want to do something a little bit different, but just to show you something. Okay, and run. And now if we click on the task in the bottom, so there's a task in the bottom, and you're going to see the task manager, and it's going to run all the individual steps. And the nice thing as a developer is I develop my logic, and it's very easy to parallelize everything here in Java. So we're using like modern Java 21, and it's then very easy for me to focus just, okay, I want to generate the extracted ion chromatograms, let's say. And I can do this for each sample in parallel, and that's what you see happening here in the task manager. And it's 21 steps that we're doing. It's already doing all the annotation, molecular networking, everything in MZmine, and I'm already done. So I hope that for you it was also kind of similar. So did everyone now, like, was everyone able to generate some feature lists? I think you might only see one feature list or two feature lists, actually. If you click on from MS data files on feature lists here on the tab. And if you were not able to, so we were in the processing wizard, so processing wizard, and then create batch and OK to run everything. Okay. So maybe hands up if you are having any issues, or did you finally get to the feature list? No hands up, so I imagine that most people are watching, or maybe they actually managed to process. Okay, and from here, similar to the raw data overview, we can just open the aligned feature list. So for you, I think you should not see like all the other feature lists, but we can just double click on a feature list and then just open it in one of those windows. And so why do I have so many feature lists now? For each file, we generate for each step one feature list. So typically, the first step is that we extract we generate the extracted ion chromatograms. So you can see here on the side, if I open the EICs, I'm going to see like all the different extracted ion chromatograms, and there might be, of course, multiple features, multiple peaks in one chromatogram that we then split into separate features. There was a smoothing step that we then did. Then we did a resolving step, so we split features in a chromatogram into separate rows in a table. And then we also applied some de-isotoping. And I got all those feature lists because in the processing wizard, I set everything here to keep. Yeah, there were some filters, and I set everything to keep, not to remove. And I think keeping is very good if you really optimize your batch. So if you're optimizing your batch and you really now want to go through and you have a feature that you were looking for, but somehow it was lost in the processing, or maybe you have a double peak, and now you want to optimize this, it's very good to then keep all the feature lists, and now we can go back into each individual step and then see, okay, here I missed it. Yeah, here I made something wrong. So then you can optimize your processing. So the question was if we already search for adducts, and it's one of the steps that we do, and it's basically user defined. So in the wizard, there's no option because we tried to keep the wizard quite lean. So we tried to not give you too many options because then we're basically just recreating this batch window. And in the batch window, you have all the steps that we did. So all the algorithms are here. And there is one step, the ion identity networking. And in ion identity networking, then you could just double click and there's some more parameters to set up. And there's also the ion identity library. And in the ion identity library, you could then basically set the M plus H or M plus sodium, potassium. There's some in-source fragments that you can search for. And there's also like multimers that we're searching for. So some compounds kind of cluster together depending on the in-source conditions. And they also going to be found here. And we can also visualize this now. So everyone who wants to follow along, like the last feature list with the longest name, this is typically the one that you'll want to look at. So it's the aligned feature list. If we look at the feature list here, we're also going to see that there's all the samples. They are color-coded. Because we have so many samples, if we now visualize something, there's going to be some colors doubled. But of course, it's quite hard to visualize. But each sample, we found some features, and they have the, like an MZ, retention time, height, and maybe MS2 spectra. And then on the row level, we have also like the median MZ, retention time, and then also the ion identity here, for example. And because we applied spectral library matching, we also got, for example, a structure. Yeah, so through spectral library matching, we already have some annotations with structures here, and those come from the MONA LCMS-MS positive library, but you can use also your own, and so on. And here's the ion identity, and ion identity really points to the ion identity that we identified for this row. And so in the table, you can always sort, for example, by spectral library matches. And if you then wander and want some additional information, there's a lot of columns that we can add to the table. So we can click on the plus, and here you're going to see that there's a lot of different properties that we can add for the feature. Currently, we only see, for each sample, we only saw the height of the feature. But we can also look at something like the full width at half maximum, for example. This might be something that is very interesting to then optimize, like go back to our processing and then maybe optimize based on the full width half maximum. And you can see here now for each sample, we now have the full width half maximum in minutes also provided. And we can use this basically for the resolving of features. And for the ion identities, there's also some additional information, so we can just click on the plus here on the side. We can scroll down and there's ion identity, and you can see there's a lot of more information. Of course, in a table it's always quite hard to decide like what to show just to everyone, because everyone's interested in different things, but you can activate any of those columns, and also you can access them either in MZmine, if you program, you could program your own module and you can basically work on any kind of data in MZmine. And we also provide, of course, export, where we export all the data that we have. Like, not all of it, right? Because it's quite hard to format everything as a text, but we export most of the data that we have and that people wanted in different text formats that you can use, like a CSV format, for example. Yeah. Yeah. So there was a question about the lipid annotations, and this dataset was no lipid dataset, so sadly we don't actually get any lipid annotations here. But lipid annotations works mostly on fragmentation rules. So we're not searching a database, but we're generating our own database basically, and then using fragmentation rules for different lipid classes. So you can set up in the batch. So this is always where you go back to. Like if you want to optimize something or change something, you go to the batch mode. And this is actually where all the parameters are set up. So the wizard is only like an surface where we only give you a few parameters, and then this is actually the processing happening. And in lipid annotation, you can then activate different sphingolipids, sterols, and different other classes, and you can basically select the different classes that you want and also provide your own classes. And there's also some sidechain parameters, so you can also kind of limit the length of the side chains and number of double bonds and those kind of things. And there's some documentation for it. Oh yeah. Yeah, so far we don't have any any standards or anything there, like any data that we base it on. So we're using like all the different databases or like the fragment rules that are described in literature. And we also do detect if there's a potential collide. So if you're searching for PE and PC and so on, and then different adducts might also then collide. And of course, if you have a double bond and then the isotopes. So this is something that we then flag in the table, and it would be definitely interesting to improve it even further. Yeah. Yeah. And you can also see we did molecular networking in here, and this is one thing that we can now go into. So I can just, in the table, I can, for example, sort by similarity score, maybe phenylalanine, and then right-click on like any node, any row in the table. We can right-click and then open the feature overview, ion identity molecular networking networks. So right-click, show feature overview, and then you're going to see something like this. So this is the molecular networks, and it looks a bit weird because I zoomed in for you with all the labels here. But you're going to see that there's directly like a feature table, the network, and annotations. And let me see. Okay. So. Yeah, and so in the networks, the first thing that we can see is this is quite a busy network. Maybe I can also find something more like a smaller network. Mm-hmm. Okay. Yeah, so just again, like if you want to open the molecular networks, I did it this way. So I double-clicked on the feature list with the longest name. Then in here, you could, for example, search for your spectral library matches that you're very interested in, or like the compound classes that you're interested in. And from here, you can then, for example, say N-acetylmethionine. Sounds interesting. You say right-click, show, feature overview, ion identity molecular networks, just anywhere on the row. And this network is actually a bit more digestible, so it's a bit smaller. And here I will describe like the different things that we can see. So first of all, the blue edges are actually MS2 similarity. So we got some spectral library match here to L-methionine. Of course, do we know that it's L? No, right? Because it's only a spectral library match. So just based on spectral library, we cannot say if it's now L or D. And also you should be quite careful with the annotations that we give you. I mean, we only give you like this was what the submitter in Mona actually says. And if you, of course, then also verify the retention time, or if you verify it with other like orthogonal dimensions, then of course you might be able to say if it's L or D. Otherwise, of course, you just be a bit more careful there. But so now we have L-methionine and then N-acetyl-methionine. And we can actually look at the spectral library matches here on the side. If I just click on any of the nodes, and we can see that with blue, there's like all the signals matching are blue, and signals not matching would be orange. And you can see that there's actually no signal that is not matching, so we have a perfect match here. We also have a quite high cosine similarity, and most of the signals are described. And now you would wonder, like, why is there a connection between these two? Okay, structurally, they're already similar, right? But show me why they are similar. We can shift, like hold shift and then click on both. And then look at the similarity. So on the top, there's the similarity tab, and similarity really gives you a mirror match of these two nodes. So again, I just click on one and then shift click on the second one. And now we see, okay, true, like there's some signals directly matching, so they are blue. We can see here, blue signals. And then there's also some that are orange. And the orange ones here are actually the ones that are neutral losses. So they also match, but they match by a neutral loss. And we can switch this over by just turning our spectrum into a neutral loss spectrum. So we flip both spectra at the precursor, and then also, like all the minuses are now like minus 17 would this be, and then we see that some of the signals now directly overlap. So you can see we have the same neutral losses of 46 and so on. And so in this one we see them as orange, and in the other one we see them as blue again. And I think here it's also quite nice that you can look at the spectrum, and then you would also get here, like if it's matched or if it's modified match, and you can see the contribution of each signal pair to the score. Sometimes I was wondering, like if you have a spectral library match, for example, there might be, for example, the precursor, which we filter out in MZmine. We filter the precursor signal out, but sometimes the precursor signal is just massive, and then all the fragments are quite small. And if you then just match these two signals, they might make up like 99% of your cosine similarity, and then you say, okay, I have a huge cosine similarity, but it was basically just a precursor signal matching, which is already a pre-filter. So be kind of careful then with the reporting. Of course, if we see that the intensity of the signals are quite distributed, so all the like there's multiple signals with similar intensities, then this is of course better because we get a contribution to the score from multiple different signals, signal pairs in this case. Also, sometimes you might wonder not only for two, but you might wonder for multiple nodes here in this network, and you can just shift-click and collect multiple spectra. And then here on the right, you can now see all the different MS2 spectra and the precursor intensity or precursor signal just marked in orange. So now we can actually see if there's like some signals, some fragment signals that are always present in this network, in this cluster. And very often, of course, because we use cosine similarity here for matching, this is the case. So here's, for example, one signal that is really present in most of the spectra, and only in one it's a little bit tiny, but then the rest really has the same signal. And you can also see that there's multiple layers. So we also have here this orange layer, which kind of connects things in a circle. And it looks very much like ion identity networking before, right? So we can go on visuals, and in the visuals, you can see that here are different options. We can just type in adduct, And now we see that there's actually an M plus H, M plus sodium, and M plus 2 sodium minus H. Yeah, so sometimes we can also see this, very much depending on your matrix in your samples and also your spray conditions. But so you can see that here's multiple ions, and if they are connected, there's different ways of connecting them in MZmine, but typically we apply feature shape correlation. Yeah, so they have the same feature shape, they have the same retention time, and now they are just clustered in a circle, and only one was actually annotated by MS2. And this blue node here might also, you might wonder what this does. It's basically like a neutral molecule node. So it's nothing that you measured. It's more derived from all the other ion identity network nodes being connected and then generating this node and connecting everything that was connected to the dot, like to the children. And there's a lot more options here to just visualize everything. You can change the color, for example, of the nodes. You can do so many different things. But then there's one option here with the filters that is quite nice. You can just go and search. So, for example, we now had methionine. We can also see if there's more other nodes that actually have something to do with methionine by just typing in meteor and then focus. And it already says there's only 2 nodes that contain this substring in the annotations. So if I focus again, we can see that here it's pretty much the same network. I can add the labels again, and now you see there's a bunch of different nodes here, and they all have different IDs and MZs and everything denoted here on the nodes now. Yeah, so if you're searching for something specific, it's sometimes quite nice that you can just go into the network and then you can just type in what you're searching for and connect it. Yes?
I have another question. For measurement like small molecules, do you have does the software provide bio ID? So for me, when I want to extract the results, I want to put it in a statistical software. For this scenario, you guys already have the statistical analysis section. Let's say I want to compare it with metabomics. Does it confront any of the raw bio ID like K, bio ID in this?
Okay, so the question was if there's like other bio IDs, like other IDs being provided. And this very much depends on the Spectral library if they provide this. But we also, Corinna is going to show some Spectral library generation workflow later. And there, for example, we have some script that already, from the structure, already gets like all different bio IDs and basically like gives you all the different IDs, clean structure and everything else. And then you could use it during building the Spectral library. But it's actually something that we should maybe also include, like just adding more mapping. Yeah, there's like great online service that already do something similar, but then you have to do it again, right? So there's Unichem, for example, where you can just give the structure and then you get like all the different identifiers from different databases. Mm. Makes sense. Okay, so this is a network visualizer, and it's definitely good to like play around, and it's a bit more dynamic. So there's other tools out there like Cytoscape, which is amazing. You can export this, and then you can like plug it into Cytoscape, and then from there you can like visualize everything more nicely for your paper, and then of course like polish it. But the idea here for us is that we want to really integrate everything in MZmine in a more dynamic way. So if I now right-click on, if I control-click on any of the nodes, I can, for example, just take out the neighbors of this one node. Because sometimes the networks get very, very cluttered, and we don't apply any filtering to the networks. So GMPS, for example, there are some like filtering options that you can turn on or off, but by default they are on. And so maybe if you know the GMPS networks, they are quite like small very often. And so just control click to get the neighbors of one of the nodes. And now maybe as a last thing for the hands-on or live demo, we also added a statistics dashboard, and this is very much in development. So how did I get there? So you can always try finding it like on the top here, but there's a lot of modules that many people have developed and contributed to MZmine. And one thing that you can do now is just like double shift or Control F. So like in any other tool, Control F to search for something, and then we're searching for statistics, dashboard. And then we can just open it by just hitting OK. And for a long time, we kind of said, okay, statistics is not our focus because there's other, like Python and R and so on, like other toolkits that are much more versatile. But then again, like it's then not connected to everything else. So here we really try to focus on all the like standard methods. So if you're missing something, please ping us so that we know that you're missing something and that we can develop it, or maybe we can develop it together, right? And bring it in here. But then we can really make it very interactive. So here we can see, for example, the PCA. We can see the different samples, and we can see the loadings of the features and the table. And just on the controls down here, I can, for example, set the coloring to the condition. And the condition I loaded from the metadata. So if you don't see it, maybe you just missed loading in the metadata. So sample metadata is on project sample metadata, or Control M. And here you can see that we had some species information and we had the condition, like QCs and blanks, and then 2 different sample types. And then in here we have some scaling that we can do. We have missing value imputation. We can set the domain principal component, also the principal component 1 and 2. Coloring, and also which abundance we use. So maybe we want to use area, and then everything is just recalculated when we change this. You see it's like automatically recalculated, and now we can, for example, click on the features here. So we can click on one of the features, and we can see that the orange features are annotated by spectral library matching. We directly get some box plots. We get the loadings from the loadings plot, and we also get the structure directly down here. And now if I had 2 screens, I could also open the network from here. So I can right-click, show networks. And now it doesn't really work so nicely, zoomed in so much. Because this you would really need 2 screens, basically. But I could, for example, let me see. I could, for example, take the volcano plot here. So on the top there's volcano plot. I can go for condition, and then the 2 different strains. And then in the volcano plot we also see everything now that is colored, has an annotation, and I would then also focus on this node in the network. So this one, for example, is a singleton, so it has no connection to anything. This one is actually connected, and you see the network, if it's on a different screen, now you would directly get a feedback and see, okay, this actually has an MS2 spectrum, and it also has some other spectra that we found that are similar in my data set, and maybe some annotations. So we try to now like basically combine all the standard statistics with with the networks and everything, make it interactive and dynamic. And this is very much from like the hands-on live demo. There's much more you can do, of course, in MZmine. There's a lot of visualization modules, and there's optimization that we did not really touch on yet. And I think I will also not do it for here, because we also have some videos that you can look for. Again, in the beginning, there was the startup screen. And on the startup screen, there's also some links to the documentation that you can read and also contribute to, because it's open. And then there's also lots of videos that we put on YouTube. And just something, because that's usually very nice. Like once you start optimizing things, there's a lot of parameters usually. Some of them are important and many others are very situational, and most of the time you can leave them. But typically you see something like this, like parameters, and then on the bottom you see show preview. And so we can just activate a preview, import some data, and when I then just select one feature list here, for example, it would automatically show me like one nice chromatogram, and maybe on the top it's actually trying to get a chromatogram that's a bit harder to resolve, or that might be just noise. And then we can basically optimize and try to optimize the parameters so that all the features we get, we get, and then that from a noisy chromatogram like this, a baseline, we don't pick so many peaks. It's always kind of a balance that we have to really set up. And this one I did not optimize, right? So I could very easily now optimize this and basically try to get this shoulder here in, and so on. Right? I can change some parameters and then it automatically changes everything and gives you some feedback. Still never going to be optimal, but in the end, I would say doing statistics, doing molecular networking, and finding something that seems like statistically significant in your data set, and then going back to the raw data and like visualizing it interactively, I think is the way to go. And then you can focus on, okay, this one seems really interesting and I should spend some time on elucidating the structure or doing everything downstream. Okay. Okay, so with this, I think I would get the next speaker, Corinna. Corinna on the stage, and she can take over. And yeah, if you have any questions, I mean, there's not so much time, like we try to have some time afterwards for more questions also, but then we're also going to be around, right, and you can just ask anything there also.
Does it work? Ah, yes. So I guess this was a lot, especially for people that are new to MZmine. So definitely feel free to reach out if you have any issue, or also there are a lot of videos online on YouTube. Then you can do it like slowly, manually by yourself. And here, maybe for some it was too fast or not the time to spend enough for some specific issues. So before I start, I'm Corinna. I'm a postdoc in Tomáš Pluskal lab in Prague, and my research is about mass spectral library building. And since we use MZmine, so I will also cover a bit how we do it in MZmine. So as you can see from the title, we acquired like 38,000 compounds in like 11 and a half days for one polarity, so 23 days in total if we wanted to acquire in positive and negative mode. Of course, it's very specific for our method that we applied, so maybe you need more, but yeah. So first of all, I want to go back one step to have everyone on the same page. So what you did now was kind of like not the statistics, but if we think about a study design, so we first have our samples, we acquire them, then we need to know which signals might be important, which signals we want to annotate in the end, and then we need to annotate them, and for this we need to have a fingerprint. And I guess all of you know the issue that so far public databases are very small compared to the number of compounds we know, or we don't know even. So this is something we want to tackle with our workflow, to have some pipeline that we can get fast the spectra out of it, have like a data file that you can use in your library or in your lab, but we also want to make them publicly available so that you're open to share the data if possible. So what can be the motivation for the libraries in general? So of course the normal ways that you have your reference standards in your lab, then you would acquire the data, you want to export or you want to extract the spectra and export them as a data file. And then of course the other option might be that you see some signals in your samples and you want to have those also in a library. And then you can, in the end, use those data to see if you see those very often in your samples. So maybe it's some contamination from your plasticware or from your sample processing, or if it's something new that you really want to discover because it was not annotated before, so it's something unknown. And you saw already that there's a lot of processing that can happen beforehand, like spectral processing, and this is like a very easy visualization what we actually do for the whole pipeline. So we start with our metadata where we know which compounds we have if you have reference data, reference standards. And then, of course, we have the acquired data. We combine both in MZmine, so to annotate the data there. And, of course, we do all the spectral processing that you just did today. So it doesn't matter which method you use. If you use GC, if you use LC. In our case, we used flow injection. That's why we were so fast. And then you do basically all the processing. You annotate it based on your metadata sheet. We also have some merging in-house in MZmine as well, so also some checks. And then we can export it in the end as different data files. We can extract retention time, collisional cross-section if you do IMS, and everything that MS2 or MSN depends how you acquired your data in the end. And the whole pipeline looks a bit more complicated, of course, so it's not so straightforward. So we divided the whole processing basically in 3 main tasks. The first one is the metadata cleanup. I will not talk today about so much into all details. There's also a preprint recently published, so you can definitely check this one out, or you can also join me at my poster number 1029 if you want to know more about it. But generally, the metadata cleanup is important if we think you get from a vendor the compound sheet, but sometimes they provide the compound as the structure, as the salt. So here in this case, we have this tartrate, so we need to get rid of this salt. So we have this as a Python script. And then sometimes you might have only a compound name and not a structure information. So we also enable in the script that you query PubChem and you get then additional structural information. And from here we can then also generate the exact mass for this compound, the chemical formula, and everything that can be computed if we have the structure in the end. There's also a script to do the sequence generation, but it's so far also only for the Orbitrap IDX, but it might be also adjustable depending on your instrument and how the sequence needs to look like for the acquisition in the end. We then have this sample preparation workflow, so it's everything that is done in the wet lab. So of course luckily we had a robot to prepare our samples, so we always mixed up to 10 compounds in one well, and then we had 384 well plates or 96 well plates, and then in each well up to 10 standards mixed. We had this dual pump flow injection method. I will also not go into detail here. And in our case, we also acquired MSN data, so we didn't stop at MS2 because we wanted to have more substructure information. So this is also covered in this workflow. But as I said, especially this step is very optional, so this is just how we did it. If you use LCMS, GCMS, it's just another workflow, but in the end you can do especially the data processing in MZmine kind of in the same way. Of course, the processing itself, it's then depending on your acquisition. And then we have our data processing in MZmine, and this I will show a bit more into details. The import and mass detection is everything that you basically did now during the hands-on session. We have then, in this case, the MSN tree builder, but it's only because we have no retention time. If you have liquid chromatography, again, it's the steps that you did today in the workshop because we need to have the feature table that you just generated. And then from here we used our local database search, so the metadata sheet, or if you have spectral libraries, you can also do the matching here and the annotation. And then we would only export those that are annotated if we use reference standards. And before we export, because it's done in an automatic way, we also want to be sure that we export something meaningful. So we also included different quality checks, and then we export it as different data files. And I said we have different quality checks, and this might be definitely something we need to optimize and we want to improve. So if anyone is working on quality checks for library data or for Spectral in general, definitely feel free to reach out to us if you have any ideas or you want to implement something in MZmine. It's definitely open to collaborate. So we now have 2 checks that we included. It's the precursor purity. So we have our MS1 data before triggering the tree and after triggering the tree. And we go back and see if we have in this isolation window something very high abundant next to our signal that we wanted to trigger the fragmentation for. So in the first case, we see it's kind of clean. We have only a very minor signal next to it. And in the second mass spec, we see we have a lot of other signals. So if you would acquire data for this, we would create chimeric spectra, and this is something we can then flag or we can remove. In our case, we normally think it's better to keep it. you flagged it, so it's also named in the compound name, so we have it like chimeric. So you definitely know that it's not a clean one, but you definitely get some information for the compound itself. And also if you think about you have like a complex samples, there's always a high chance you will generate in the end chimeric spectra. So it's also good to just keep it and you know there is information, but also additional information for something else. The second step for the checkup is like a subformula notation, because we know from the compound table which chemical formula we have, so we know what is the starting point. And then we see if we can explain each signals with the subformula. So we don't take the structural chemistry into account, so it's really just the chemical formula, and if we can mix it in any other combination up to the maximum number that we started with, if we can get the same signal or M over Z to explain this signal. So this is also checked before, and then you can say if you only want to export spectra with a high explained intensity or number of signals. And of course, you can also export everything on the first hand, and then it's still stored in your library file itself. So you all the time see those parameters still in your table or in your file, and then you can, remove them later on, but you still keep it for the first run. And this is then the feature table that you get in MZmine. So we have here, it's the data processing, what you today did. And this is the annotation. It's basically kind of similar to spectral library matching, what you did today. But in this case, we only check if we have the M over Z, and if you can explain it with something that should be included in the sample with different adducts that you can also enable in MZmine. So you start in your compound table with the exact mass, and in MZmine you can say which adducts you want to include, and then based on the calculation and based on the file that should contain those 10 compounds in our case, you can then search if there's any mass matching to of your set compounds and adducts. And speaking about it, so this is like a plate we analyzed. So we had like a lot of plates. We acquired them in 11.5 days, as I said before. And we also wanted to see how many of those spectra we can actually see already in the public domain. And this is just like a small subset that we used. So it was around 50,000 MS2 spectra. And then we see here each node is like one MSMS spectrum. And we try to distribute it in the chemical space based on their similarities. So nodes that are closer to each other should share some similarities. And we see only 14% were annotated, so everything that is colored in magenta was annotated. And we see it's a very low number of signals that was matched based on this. And if you look even further inside, we can also use feature-based molecular networking, what you also did in MZmine today, and we see that we have some clusters corresponding to a specific compound class that is highly covered, like the beta blockers we see in GMPS, which is like the biggest public database so far. We have a lot of compounds, so we would annotate this, or we would be able to annotate this cluster even though we would not know anything about it. I mean, in our case, I know about each spectrum, of course, what it is. So this makes sense because it was the beta blockers. Then we have the sartanes. We see we only have like 3 annotations, but this might give us already some hint what the cluster might be corresponding with. And then, of course, you have clusters that are totally unknown, and you would not know anything about it. In our case, this was for the cephalosporins. So if we would not know this was our reference standard, it would be blind what we want to look at in the case. And this is something I want to stress out. It's a worldwide effort, so everyone who's acquiring reference standard should definitely be open, or if possible, be open to share your data publicly. And you see we have a lot of, the world is big. We have so many groups working on so many different topics and research fields. So that's why definitely if you acquire reference data, feel free to upload them and share them with other people. And this is just a very quick update. So this is the data that we now, or the libraries that are now kind of available. Of course, there are many more smaller libraries as well where you can get data, but I used the 3 most used public databases like MassBank North America, MassBank EU, GMPS. Then we also included MZ Cloud because it's the biggest MSN library that we have, and then NIST 2023. But for the last one, we have the problem, they are very big and they are manually created very often and from high quality, but the data can't be downloaded and used for any method development for machine learning, so we are limited. We can use spectral library matching if you have the software, but if not, you can't use it for anything, so it's not yeah, it can't be used in the future. Because if you think we can only annotate things that are in the spectral libraries, we are still very limited what we can annotate based on this. And with our effort, we see that from the 38,000 compounds, we could detect like 30,000 combining positive and negative mode. And we get like more than 2 million MSN spectra out of it, which I think is already showing what we can actually get. And in our case, we use flow injection, so super fast. So that's why we only needed like 23 days. But it's showing how fast we can actually increase the databases in the public domain. And this is just, again, some statistics. So I want to highlight why positive and negative mode might be interesting for you. So it's good to see that a lot of compounds were detected in both ion modes, but still we had like more than 2,500 compounds that we could only detect in negative polarity. So really depends what is your compound of interest. We need to be aware of what we can only detect if we only acquire in one polarity. And then if we see this statistics here, it's about how many unique compounds we have in each database. So we see if we get the biggest one here, which is open, and with open I mean Mass Bank North America, Mass Bank EU, and GNPS. So we see we get like almost 25,000 unique compounds that are not included in any other database. And if we look here to this is the MSN lib, we are almost reaching the same level just in our 23 days, just acquiring data in one lab. And this is definitely something we need to extend again. And I think this shows you what we can actually reach as a community. So we definitely want to increase the annotation rate. If we have more data, of course, we can already do more spectral library matching and annotate more. But one issue still, we need high quality data to train models with. So we know Sirius and MS2 Query, for example, they use spectral libraries for training their models. And also if we have MSN data, which is also so far kind of limited in the public domain, we also can use it to annotate substructure fragmentation or to actually learn more about fragmentation in general. Yeah, and I think with this, thanks for your attention, and definitely if you have any ideas, if you want to collaborate, feel free to reach out to us.
Thank you, Corinna. And now we're going to have Roman present in his machine learning effort. And just a brief finish from my side. You all maybe are a bit overwhelmed with the hands-on session, and that was pretty much expected because it was very fast. We only had like very short time. But the one take-home is set up your batch mode with MZwizard. So really try to optimize it there. And then once you did that, save all the parameters, save everything that you've done in the batch mode. So you save this batch, and you're going to get like an XML file that you can also upload with your publication. So once you publish, you can upload this on any like repository with your data, for example, or with the paper itself. So very important that other people can reproduce your results. And now, Roman.
Yeah, so hello everyone. My name is. Can everyone hear me? Okay. So my name is Roman. I'm a student in Tomáš Pluskal group, and I'm going to present the main project of my PhD, which is where we develop a new universal machine learning model for the interpretation of tandem mass spectra. So what does it mean? It means that we have a single neural network, which we name DREAMS, which given a tandem mass spectrum can make many predictions about the corresponding molecular structures. So for example, it can predict molecular properties or the presence of fluorine or the molecular complexity, molecular fingerprints or spectral similarity between a couple of spectra and so on. So it's easy to extend it even further. Whereas if we consider existing methods for all these tasks, we can find that for each of these methods there exists a separate tool, and if this tool is good in one specific task, it doesn't necessarily generalize to any other task. And I will not go much into details, but in our recent preprint we show that our method is not only universal for all these tasks, but also outperforms each of them. And throughout the slides I'm going to show how we achieve this. But first I want to mention why am I even presenting it here. Because during the summer we are planning to integrate DREAMS into MZmine. But if you want to try it out even today, then you can check out our GitHub, and you can access the neural network via an easy, via a user-friendly Python API. So to begin with, the annotation of MS spectra is inherently a very challenging task. So if we consider some arbitrary LCMS run, we can find that it's super hard to annotate all the spectra, and in fact, the vast majority of them remain poorly understood. And one possible reason why it is so is can be found in the following way. So historically, from the 1970s years, the most dominant method for the interpretation of tandem mass spectra is simple spectral library lookup, where given a new experimental spectrum which we want to annotate, we simply compare it to reference library standards, and if we find a similar spectrum, we conclude that the corresponding spectrum also has a similar molecular structure. And you may probably know more recent methods such as Sirius or MIST. So, and even though they are based on machine learning or combinatorics, and basically they are much more advanced, they still rely on the same spectral libraries as a training data. So training data is just essential for machine learning. But there is a severe issue with spectral libraries. If we perform a simple analysis of the composition of molecular structures, we can find that they don't even cover 10% of, for example, known natural products, or they don't cover human metabolites. So it's very hard to imagine a new model trained on these spectral libraries which can generalize to completely new unknown structures and to make predictions about them. So this is our reasoning why this annotation is so hard. But what does exist is a huge repository of up to 7 billion raw mass spectra. And the bad news about it is that it's completely unannotated. We don't know any molecular structures or pretty much any information about the corresponding spectra. But the good news is that this data is really huge and it's diverse, coming from diverse biological and environmental studies. So the first thing that we did in our project is that we refined these millions of mass spectra into a high quality and non-redundant subset, which we name GEMS, GNPS experimental mass spectra. So, and once we have all this data in hand, the obvious question is how can we even leverage all this data, or how can we extract any meaningful knowledge from it? So, and our answer is that we can apply the paradigm of self-supervised machine learning. So you may probably know it from the ChatGPT. So as you may know, the main training objective for ChatGPT is simply filling in missing words in millions of text examples. So in our case, we do pretty much the same, but instead of training the neural network to fill in the gaps in text, we train the neural network to fill in missing peaks in mass spectra. And I will not go much into details here, but I want to emphasize that our approach can be easily scaled up with all the data we have in hand that we collected in advance. And also we can easily scale it up in terms of the neural network size. So, and what we observe once we scale it up to these millions of examples, that the neural network kind of discovers molecular structures just by being trained only on the mass spectra. So, and why do I say this? So it is kind of represented by the following figure. So each point here is a tandem mass spectrum, but it's not just a tandem mass spectrum, it's how the neural network see it inside. And so each representation, each of these points is in fact a point in 1000 dimensional space, but here it's just reduced into a plane. So, and we call this representation dreams, deep representations empowering the annotation of mass spectra. And you can see here easily that even though the model was trained just on raw mass spectra, by the colors it kind of separates well all the spectra by the structural properties of corresponding molecules. For example, carbohydrates are well separated from halogenated molecules and so on. So this shows that model, to some extent, model understands chemistry behind mass spectra. And even if we go one step further, we can do exactly the same analysis, but not for where each point is not an individual mass spectrum, but a single LCMS run. So we basically just average these representations across runs, and we can also see that the model captures for example, the taxonomy of foods when analyzing a food data set, but it has a much bigger potential. For example, we can start comparing species between each other or perform more biologically relevant analysis. So what I have shown so far is that model understands molecular structures behind mass spectra, but how can we achieve the state-of-the-art performance on the 5 tasks, as I showed at the beginning, is very simple. We can apply so-called transfer learning, or we can basically just slightly fine-tune the model to go from these dreams representations to specific tasks, such as the prediction of molecular fingerprints or to detect flora and so on. So again, I will not go into details. Yeah, so these tasks, as I mentioned at the beginning. And just to summarize everything, so we also develop a publicly available resource called Dreams Atlas, which just combines basically everything I have shown so far. It's a huge molecular network for 200 million MSMS spectra from massive GNPS, which is constructed purely based on dreams. And, just to give you a flavor what it contains, so if we just zoom into one specific node in this molecular network, we can see that it contains a lot of interesting information. So each point is one of these 200 million mass spectra, and it also contains metadata from massive, such as the sample, the species of origin, or the description of the study. So if we, for example, zoom into arm psoriasis study, we can find the following spectra in the neighborhood. So we can find that it's somehow connected to pesticide. So we can, and it's also connected to different environmental samples. So we can conclude that a possible hypothesis for the emergence of psoriasis is, for example, that it's provoked by the connection to azathiostrobin pesticide, so which hasn't known before, for example. Yeah, and thank you for your attention. If you want to chat about this, or if you have any questions, please let's talk at my poster 1048, or feel free to check out our preprint and GitHub.
Thank you, Roman. And Louis, are you ready? So I want to highlight that Roman is doing some great work with machine learning, and like the PyTorch model that he's been developing is like out there, so you can use it directly. And also in MZmine, we can very easily now like integrate it, because there's a like very nice library where we can just integrate any PyTorch, also TensorFlow model in MZmine. So also if you're working on something, that might be like a great way of also getting this out there, not only developing the model, but then also serving it to like a wider community.
Okay. Mm-hmm. The microphone is this one?
Yeah.
Okay. It's showing up? Okay, so hello everyone. In this next minutes, we will present you what we can do with large language models to facilitate the learning of advanced softwares like MZmine. As you might know, the MZmine community has been very active for several years, and we have on the web, in the public domain, research articles, as well as workshops recordings, many from the colleagues that are present here, and the GitHub repositories that are listing the issues observed by the community, as well as some recommendation about how to solve some some bottlenecks and so on. And one list of these resources is presented here. So we have the video recordings, the GitHub repository, the articles, and so on. So what we have been doing is creating a chatbot, just like ChatGPT, that has access to the knowledge base of the MZmine community. So initially, all the files that you are seeing here in this list, so YouTube articles and GitHub repository, all of that is assembled into a knowledge base that is vectorized in a database that the large language model, so I'm using either Mistral or ChatGPT for the demo here, is accessing to retrieve some context that is relevant to the question of the user. So if you have tried ChatGPT on your research project, you might have observed that it doesn't know much about detailed softwares or even less about the experimental data. So here we are providing some information, context, that will help the large language model to provide some meaningful answer. And it's my pleasure to present you a quick demonstration of this. Okay. So I will just restart everything. It's running on my computer at the moment, but it will be soon available online. Okay, I need to check this. Okay. Okay, so this is the interface to chat with the MZmine knowledge base. And I will just start with a question, but I would like to have someone or several people challenging the bot to see the performance. So the first question is using Mistral AI that is less performant than ChatGPT 4.0. So I will give it a try. So what happens here is that from the input question, the model is extracting from the database a set of documents from the knowledge base that we have assembled. And okay, so here, 5 documents were retrieved from different sources. So we have this preprint article, the GitHub repository from MZmine and GNPS. And you see this is the text that was generated. So is it consistent? I'm looking at it briefly. Feature finding optimization. Okay, so it gives some information about the software and how to resolve or to optimize those parameters. It's a very open-ended question, so it might not be the best example. So I'm opening the floor, the mic, for someone that would be willing to give it a try. It has to be related to MZmine, though. Okay, someone. CMS, LCMS, IM mobility. Okay.
Hi, thanks. Is it possible to integrate data from across different chromatographic batches with MZmine?
Is it possible to integrate different, you said?
So data from different chromatographic batches.
Chromatographic batches? Okay, I'm not sure I understand the question. Chromatographic batches from different data sets of different...
of different assays.
I mean,
with like internal standards, obviously. But
I mean, chromatographic batches is not a standard denomination.
So you mean like multiple sample batches? Maybe like integrate multiple samples acquired in separate batches. Yeah. Yeah.
Okay.
Yeah. Close.
So the question is very complex, and if you allow me, I will just switch to GPT-4o. That is a commercial model that performs better. So I'm retrying with the same question, a different model. So already the formatting of the output is of higher quality, And usually the content is also better aligned with the user question and document. So it seems consistent: data import, pre-processing, alignment, gap filling, feature filtering, export integration, and so on. So an important part of this chatbot is that we can track the original source of the information. So one might be interested in looking at this article, but also at some documentation provided on this website. Okay? Anyone else is inspired?
Maybe. Oh, someone? Okay. Yeah. I had a question.
Is it possible to process DIA data? DIA?
Tricky question.
The answer is no. And I would like to ask Robin if it's correct. Can we?
So we do have like a experimental workflow for this, so it's quite new. And so it kind of makes sense because maybe the answers are also sourced from the issues. And in most of the issues, we then point people to other software and saying that maybe use this software now. So it's also some like old information sometimes. So we would also need to see like the other. And yeah, like DIA, we're really just integrating now more and more.
Okay, so that one was a success, let's say. Huh? Anyone else? Doesn't work always, so I'm glad.
Yeah, so is it possible to process the MRM data in MZmine?
Oh, I need to clear the discussion, otherwise it will mix the two questions. Okay. So it says not straightforward because it's mostly for untargeted metabolomics. Okay. Precursor product ion transition. Okay, so again, is that correct?
It's kind of correct, yeah, but I think the MS Convert software also allows to extract it into Spectra, and then you can. Okay, yeah, maybe, yeah. So we're definitely working on this like in the future, because that's also something that many people want to integrate then, maybe even with untargeted and targeted data, right? And then, yeah, so it's not very straightforward. So there's some ways to convert, but not so.
Is it possible to use? Is it possible to process ion mobility data, especially multiplexed?
Multiplex ion mobility data?
Yeah.
Like, what do you mean?
The way it is collected in Agilent 6560 or Mobile Ion.
Okay. It says yes. I don't have the answer myself, so please, if you can comment.
So yeah, so we do integrate, for example, from timsTOF, we integrate the native format. We're also working with Agilent to also integrate the native format so that you don't need to convert. But so far, you can convert the data to MZML, and then I hope that also the multiplex data, maybe we talk afterwards, because I hope we can integrate that already, but we're also working with Mobile Ion to also get their data supported. So, but in the future, we want to support it like natively. Yeah.
So thanks a lot for the questions, challenging ones.
Maybe I have one question.
Yeah, go for it.
So maybe you can clear again, and then what is the difference of IIMN and FBMN? So ion identity molecular networking and feature-based molecular networking.
So it's GPT-4o.
Let's see if it's
running.
So it's two different. It's basically ion identity works on top of feature-based molecular networking, and so maybe interesting to see if it's...
That I'm not sure. It
No.
kind of hallucinates.
Yeah.
Yeah. The rest is okay. Feature-based. Robin Schmid here, good. Application, key differences.
Ah, okay, yeah.
Application. Yeah.
Okay. No, looks quite good.
Okay. So this is just an overview of what can be done, and my key message here is that the knowledge base is the key in order to deploy such AI capability, because the documents, they have to be in the public domain. So any YouTube recording, like a workshop that is on the web, can be used for that. Similarly, preprints, everything behind a paywall cannot be used for such applications. So again, my message is that if you feel like some of the content you are generating can help the community, make sure it's available online, because then we can bring this in such a system and deploy this at scale for everyone. So this is on my computer, but in a few days it will be released on the web, and any one of you will be able to use it for free with the Mistral LLM model that is it's free to use, unlike GPT-4. And we'll be able to search the GitHub repository, the many recordings available on MZmine, feature-based, and so on, as well as the research articles. Okay, that's it. I'm available for any question after this presentation. So thank you very much.
Thank you, Louis. And that's also important why documentation and all the other resources that we build are so important, right? Because then it's like findable, and we can really reuse that. And again, the documentation of MZmine, also GMPS and other tools, they are usually open. And that really means that you can not only consume them, but you can also add to them, right? So you're the experts actually using the tool. And so it's sometimes good to know, like, oh, there's like a little issue, and I can like add some information so that people can like really get faster to working. We have like a few minutes, and so maybe like we can get all the speakers up, and you can just ask another question, and basically it's only a few minutes, and otherwise we're going to be here also. So anyone has some questions that might be interesting for like the audience as well?
Hi. So my question is about MSE data from waters products. Is MZmine also able to process those kind of data?
The short answer is it's not very well supported at the moment. A little bit longer answer is that we are working very hard on having it supported, because there are many people who are using these MSE or data-independent workflows. So yes.
Hi, it's a very basic one. Can you import process data into MZmine? Like process data somewhere else and then import to do identification and further processing?
You can... Well, I'm thinking we had at some point an import module for MZtab format, right? So you could import the feature list from MZtab, which was created by another software. Is it still there in MZmine 4?
No, we're just updating this with MZmine 3. We made some changes to the data structures, and so like going forward, we want to support it again, like, and then from the MZtab format. But we're still working on this, and maybe also from like a CSV format, so that you can just give like boundaries for retention time, maybe also iMobility. It just comes with like the multi-dimensional space that we're covering. It's sometimes a bit tricky, But yeah.
Thank you.
Hi, I just wanted to clarify for the library building, the data quality checks that you're doing with checking for chimeric spectra. So MZmine is automatically doing that. Is that right? You're not doing it manually?
No, you just do it in MZmine, and the nice thing is that you export in the end all quality checks with the score as well. So you see what was the purity for the precursor, what was the explained intensity. So everything is already done in MZmine automatically.
Okay. And when I do a normal data set, will it give me a score? Will it do the same quality check in, say, a DDA experiment?
Yeah, in some ways we also do this. So there's, for example, like an intra-feature MSMS similarity that we calculate. So if you have multiple samples, for example, and then MS2 spectra in different samples, we compare them and also give you then like the maximum and medium MS2 similarity between the features. We do export this. There's like one format, like the CSV format, where we export everything that's in the table as far as we can convert it to some string format, right? So you're not going to have the chromatographic shapes there. Yeah. But it's not all there. And if you're missing something, it's quite easy to add it. And so if you're really missing something, just reach out. It's also easier as a developer to go in and basically grab any data that you need and then to make your decisions, right? So
we just have a short question about sometimes we are not interested in the compounds per se, but to the chemical family. And we'll be able to create, like, I don't know if it's an option to create a network with in silico fragmentation pattern, for instance, like comparing with Sirius, because we spoke about Sirius. And I don't know if it's feasible, it's just a question.
I just answer what can be done easily is that you can export data from MZmine for Sirius annotations, and then you can use all the Sirius modules to annotate, for example, the compound classes and so on. What we don't have yet is importing this annotation back into MZmine so you can use it in the context of molecular networking and so on, but we are working on it. Yeah.
Thanks.
Yeah, we're working with a team of Sirius, and yeah, we're going to import everything back and also directly hit the APIs from Sirius in the future. And networking, if you have a different way of connecting features or chemicals, we can also import like a very generic edge file and then, yeah, like show this in the interactive visualizer. We only have time for one question, and then we have to sadly end the...
Okay, thank you. Is it suited for the single cell metabolomics? It's for a direct sampling without a chromatography.
So single cell analysis with direct sampling, I mean, there's many different sample introduction systems, right? And we try to get them all in there. So we already have some multi and some like spot analysis workflows. And very often, if your analysis is similar, so like a spot analysis where you only have one spectrum or maybe multiple spectra in a row per sample, you should be able to even use the same workflow then in MZmine. And if it's somehow missing, we can also add new presets. So we can add new presets to the wizard so that everyone else also finds it very easily and can then like, yeah, start processing very easily. Yeah, thank you. So maybe, yeah, thank you so much for the session. It was very fast because only 2 hours. There's more workshops to come, so we don't want to take time from them. We do have badges if you want, some MZmine badges. It's all up to you. There's some stickers, some are transparent, but somehow there's still like this little outline out there, so maybe you have to cut it. But then there's also white ones, and if you don't want to run around with the MZmine sticker, okay, I put my laptop away. There's also the MZ stickers, so just to be like a nerd and say that we're team mass spec, right? NMR is cool, right? But we're team mass spec.